I want to thank you for inviting me to speak today. This is the first time I'm speaking about the work here. We're just over a year old at Summary, so thank you. Today I'm going to be talking to you about paediatric brain cancer. So just a very brief overview. The most common paediatric solid tumour we see in children in Australia, and 1 in 4 children cancers are brain cancers that we see in Australia every year. This is just a graph of relative survival by time period. So as you can see, with other cancer types, there has been improvement, and this continues on for, I know this is quite old data, but we're seeing the same trends in the last 10 years. And in the last 30 years for CNS patients, we have not seen a great improvement in long-term survival, and so we need to be doing better. So introducing the Paediatric Neuro-Oncology Group to Summary, the cheese grater most people know us as, and we're the newest cancer group to join the precision cancer medicine theme. And we focus on CNS tumours and understanding the molecular basis and interplay between molecular and clinical factors for our patients. We strive to have better outcomes for those affected by CNS tumours, and ultimately we want better survivorship outcomes for the patients. And to Tessa's talk leads straight into this. Our first port of call was ethics. We also didn't have too bad of a time with our HREC. They were really supportive of the work we wanted to do between Summary and the Women's and Children's, and so yeah, we didn't have too hard a time, but it was a lot of paperwork. So, and what we want to focus on is creating South Australia's first paediatric brain cancer biobank. So we have established adult in South Australia, but no paediatric. So any patient coming into the hospital in the paediatric sense, the samples are being used for diagnostic purposes only and not research. So to explain, biobanking is the collection, processing and storage of biological samples and data for research. Samples are generally kept indefinitely, and sometimes for several years based on the study. It depends on how much permission we have to keep them. And increasing the availability of these high quality samples has the potential to advance global health, and biobanks really facilitate domestic, international and national sample sharing. And so introducing to you the Brain Tumour Biobank Assay, Brain Turbo Assay. We ran it through a lot of acronym, Adelaide's very full of acronyms, I find, so we tried to think of something snappy. So, centres Australia-wide and internationally have access to retrospective and prospective access to the samples for biobanking. And up until September of 2022, at the Women's and Children's Hospital and SA Pathology, we had none. So there's 2 ethics we have and 2-pronged approach. So a retrospective access to tissues, so historical tissues that have been stored at the Women's and Children's for up to 80 years from biopsy or resection patients that are no longer used for diagnostic purposes. So we gained ethics to be able to use these samples, and some of the examples are formalin fixed embedded, the solid tumour, slides and snap frozen, and also clinical data. And then our new patients that enter the hospital are consented for a lot more tissues that we can take into the biobank and use for our research purposes. So just a quick overview of the process of biobanking. So when a patient enters the hospital and is diagnosed with a suspected CNS tumour, myself or one of the clinical team will consent. So we talk to the parent or guardian, explain what we're doing, and that during surgery, any leftover product that's not used for diagnostic, nothing extra is taken from the patient except for a small blood draw. That if they would be happy for us to take that into the biobank for research purposes. And then once the patient's in surgery, I'll wait around for a long time often, and for tumour tissue, blood and CSF, and they can consent to all or none, or some just want the tumour tissue, some want nothing else, and that's fine. And then I take it back to the lab and process it, and my job is to basically take a small amount of tissue, small or large, depending on what I get, and annotate it into many different ways that can be useful to any researcher that wants any type. So trying to do the most with the leftover tissue so that it can benefit researchers. And then it's used in, and then it's stored. So lots of different storage methods, like Tessa mentioned, in liquid nitrogen, we store a lot of our samples there. So our progress so far since September of 2022, we have over 1,000 specimens now, and that's largely due to the biopsy and resection, the older samples. And we've been newly banking samples, like I said, since September. Now, one of the major jobs has been reorientating the samples. The older samples are in a wax, and it's useful, but it's not, you need to re-annotate them to be able to use them with today's standards. We're way better at doing things than we were 80 years ago, so that's been a massive job. And as you can see, the first picture is how the old samples, they're in paraffin wax, but wrapped with a patient identifier and in sticky tape, basically. So my job has been to melt them down and re-annotate them into these, which are de-identified, no patient information is ever put on them. And this allows the histology department, for example, to cut slides easily, so that, and the ladies had lots of examples of how they use those samples. And we've also already shared some samples with our team in Perth, a very rare brain tumour, which we were able to put into a mouse model to hope to grow, to try different methods of treatment. So, and we've also been involved with clinical trial P022, which is for the DIPG patients. It's another treatment type. So our next steps for the Biobank, including continued collection of samples and clinical data, is to expand our capacity within the lab through our work with people in Australia and then worldwide. We want to grow up some organoids. I might be speaking to you about that because I don't know how to do that. So xenografting, so getting fresh samples straight from surgery and putting it into a mouse model to grow it. So that we can do drug testing, many different things to see what we can do to help the patients. And an MTA agreement, it's more paperwork. Basically, if you send a sample anywhere, you have to have an agreement between the institutes and with the lawyers, it goes back and forth. But that's something that we work on in the background so that when we do have a sample, we already have that in place and we can send it freely. And so a review of the retrospective sample diagnosis. So as you can imagine, historically, we're learning more and more things as time goes on. So a lot of the diagnosis of the older samples may not be quite correct. So we're reviewing them with our neuropathologist to see exactly what these patients have had. And we want to, there's no use in collecting all these samples and not being able to do things in the lab. So that's, and then we're continuing to offer clinical trial support, mostly in the way of lab support. So a lot of the clinical trials, people are very good at what they do, but then don't have as strong a lab background, so they send it to someone like me to help out. So these are our collaborations worldwide so far. So we have a strong collaboration with Canada, as I've come from there, been working with brain tumour research for 11 years there, and the States, Europe, and then Australia-wide. And I can't stress enough how thankful we are to our funders, especially the NRF. We couldn't have started the very first SA Paediatric Biobank without them. It takes a lot of money to do what we do, and it's so important, and we can't thank you enough. And I don't want to skip over the funders, but I did want to give people an opportunity to take a picture. So SAMRI also offers public tours every Thursday at 10 a.m. I've heard it's booked up till August, but I have been told that I can potentially have a private tour organised if there was enough people interested. SAMRI is really interesting, from the outside, but also what everybody does inside is equally as interesting. So I would be happy to try to organise a tour for anyone. I'm taking a couple of the girls through next week and to share more and more about what we do and what we're about. So, and that's my email. If you have any questions at all, you're very happy to answer them.
Thanks, Amanda. Any questions for Amanda? While we're thinking about that, so we have, I talked about the BCCC building, 2 doors down, coming up, but of course in the basement of that is Australia's, the Southern Hemisphere's first proton therapy facility, which will be used to treat children with brain tumours. And I guess, do you want to just talk a little bit about the importance of that and the work that you're doing?
Yeah, so our current treatment methods for radiation is radiation with a lot of the patients, and the issue with that in the brain, for example, is this is how I explain it to my family, radiation hits the tumour and then continues on into the normal brain, and obviously that's not good. So, and especially with our paediatric patients, often radiation is not an option for them because of their age and size. It's just too damaging to the brain. So the proton therapy is so important because the people that are a lot more intelligent than me, they program it to hit the very outer edge of the tumour, and then the radiation drops off. So it saves a lot of the normal brain from having damage from radiation. So it is, I'm so proud as an Adelaide girl that it's coming to Adelaide because it's such an important diagnostic tool and in the clinical scene. And I think it's really awesome. I think we should be really proud.
And I think also that nearly every child that we have in radiation therapy will be coming to Adelaide to have it for brain tumours.
Yeah,
and so that really emphasises, I guess, the work that we're doing in terms of the biobanking as well, because it actually makes Adelaide as a focus for paediatric brain tumour biobanking as well, because all those kids will be coming here.
Yeah, I mean, we can't. The ladies that spoke so nicely before, the samples they're using come from biobanks generally. They have to, the consenting process that comes to a biobank, and so we really can't do the work that we do without the patient samples. It's so important. You can only do so much with images from an MRI, etc. So Adelaide is, and this is why we're setting, with the help of Stuart and his team, Jordan and I are setting the biobank up now way in advance so that we're ready to go as soon as these patients start coming into Adelaide. So, yeah.
And one of the questions I often get asked about biobanking is, why do you need biobank? There's biobanks in the US or in Sydney and whatever, but all of these tumours are different.
Yeah.
So we need lots and lots of different patient tumours to be able to understand all of the tumours as a group, right? And so we may end up being able to understand a therapy for one particular group of patients. It might only be 10% of the patients, but we can, once we get more and more samples, we can start understanding different therapies that might be relevant to different patients, and that's the importance. So even DIPG or glioblastoma, they're not the same, right? They're all glioblastoma patients do not have the same tumour. They have different mutations and may be treated down the track with different therapies. That would be the hope.
Yeah.
So.
And a lot of the time, if we might see one DMG patient a year, so if we're banking that, and then as a whole, we can't do much with one sample. So as Chloe said, so if a collaborator in the US reached out and said, we have this great research underway and we need all your DMG patient samples, and they'll do that all across the world, and then they can create a pool for what's a very rare tumour. They can have 50 samples versus just our little ones. So
because as researchers, what we don't want to do is do all of our work with one patient sample, think that therapy is going to work for all patients, and then get into clinical trials and realise actually it was only like 0.1% of all patients might respond to that therapy, and we just happen to get that that one patient out of 1,000 that would have responded. So we need to do lots and lots of different patient tumours to be able to understand.